Okay, we better get going with our last speaker, Todd Woodward, from my department, the Department of Psychiatry. And yeah, it's a pleasure to have our last talk focused on human functional imaging in human disease. So, Todd, thank you.
So, Ryan Lim, where's Ryan? Oh, okay. You want to come down here? So Ryan and I are, well, I studied at the University of Victoria, and Ryan did as well, actually, but we worked together in my laboratory at Women's and Children's Hospital. So the questions that are up here are the questions that I always became interested in from the first days I started thinking about the brain and what the networks are that are involved in the brain. And at that time, there was no neuroimaging, so we had cognitive psychology at the University of Victoria, and I thought that was great, but there was no brain. We had neuropsychology, which was great, but there was no tasks. You had to just have paper and pencil tasks, and you weren't allowed to change them at all. So I just bailed on the whole thing and studied statistics, because I figured that would be helpful too. So then when brain imaging started, that is the perfect combination of cognitive psychology, neuropsychology, and multivariate statistics. So then I finally thought, wow, I can start to try to answer these questions. What are the anatomical characteristics of task state functional brain networks? What are their cognitive functions? What extent are they task specific? So when I got to the field, it turns out that for people that were studying tasks, they actually weren't interested in networks at all. It was all univariate analyses. I'll explain that in a second. So then finally some people started studying networks, but they were only interested in resting state. And to this day, there really aren't any fMRI labs that are consistently studying networks involved in tasks. It's starting to come into focus now just in the last couple of years. So we've been 15 years on it, so I think our timing's not too bad because it took us 15 years to get anywhere. So here is what fMRI data actually looks like. And so this task, now the problem, of course, for those of you that are not into cognitive neuroscience is that you have to fool around with these tasks all the time and try to follow what people are saying, what the tasks involve. So I'm very sorry if you're not interested at all in cognitive psychology, but if you're going to be dealing with task-based fMRI, you're just going to have to learn to suffer through these tasks and the explanations of what they do. And that's going to be some discussion on that today. Apologies in advance, but we're not going to be long. So this task involves looking at 4 letters for 4 seconds. Then for 4 seconds you see nothing, and then for 1 second you see a letter and you just say yes or no whether you saw that letter. There are 4 letters or there are 6 letters. Sounds pretty straightforward. The thing is, every time you do a task like that, there's a number of networks that are involved in completing that task. So it's not just a straightforward type of this brain area does X or this brain area does Y. Entire networks get elicited for all tasks. So you probably recognize those networks. So some networks deactivate when you do a task and other networks activate. We all have probably familiar with the default mode network, which is the one that deactivates when you start to carry out a task. And there it is here. And these other task-positive networks, there's a number of different networks. Now, the ones that are involved in tasks are actually not the same that are involved in rest. Everybody wants to know the difference. I don't really care that much because I've only really been interested in the task-based networks, and to explain how they're different from resting state networks is not a very exact type of explanation. But, so I'll get into this later, but the thing is that the real question, remember the things that we wanted to know about, anatomical characteristics, okay, that's actually not that difficult, but what are their cognitive functions? That is the difficult part. What do they actually do? Notice if you compute networks and resting state data, you don't have any idea what people are doing while they're resting, because they're not doing nothing. We all know that. There's a lot of stress being in the scanner, thinking of what they have to do, trying to, I don't know, keep their eyes open or closed or whatever having people do. But we don't have any timing information about those things. So we can't really, even if we get a picture, you can't say for sure you know what that area is doing. We say, oh, it's visual cortex. They must be involved in vision. Or it's dorsolateral prefrontal cortex, must be involved in working memory or some other type of function that's speculated. But with task-based networks, we have the hemodynamic response function. So you may know that those of us that study functional MRI are stuck studying the blood oxygen level dependent signal. We aren't able to measure the real-time neural activity like you do in neurophysiological measurements or EEG. But we have to study the blood flow. So there's a ratio of oxygenated to deoxygenated blood. When neurons fire, blood flows to the area. More and more blood flows with oxygen to the areas that are most active. So when we interpret these, now it turns out that these networks all have slightly different timing, and it's the interpretation of the time that the activity happens and whether or not there's differences between task conditions that help you assign a function to each network. So this network, for example, we call response, and we know that because there's one peak here and a second peak there. And so sometimes in this task there's 4-second delay, but in other trials there's a 0-second delay. So if you don't have one peak here and one peak there for the condition when there's a delay and when there's not a delay, you don't have a response network. It's not really enough to just look at the neuroanatomical explanation and say, well, this is left sensory motor regions. We have some activity in the right cerebellum. That's not really enough to conclude that that network is involved in response. You have to, first of all, show that the peaks of the hemodynamic response shape are in this right spot. And in this situation, you shouldn't see a difference between 4 and 6 letters, because by the time you're responding, there isn't a difference between 4 and 6 letters. That only matters when you're encoding and remembering the information. And that is the case with the response network here. So we're comfortable saying that that is the function of this network. The other ones are not as straightforward. We'll see whether or not I get time, but you can get some hints by, for example, looking at the peak. So this is seconds. We have to study like 20 seconds worth of time to get the signal from fMRI. So here we have 10, 12, 14, 16, 18, 20 seconds. Okay, so the peak for these, this is about is that 12 seconds, 16 seconds, I think. I can't really see it that well. Oh, wait, no, I do have a display here. I can't see that either. Okay, but the peak of this peak is earlier than this peak, right? So this is involved with visual attention, but these other networks, the default mode and the what we call internal attention, are actually peaking a bit later than the visual attention. So you see something, you pay attention to it, some stuff happens, and notice that during that time, there's a big load effect. So whatever's going on between seeing and responding is definitely more active or deactive for the default mode when you're seeing 6 letters compared to when you're seeing 4 letters. So it doesn't really take a rocket scientist to start realizing this is involved in maintaining that information online for when you have to respond. But the thing is, the same network comes up in all kinds of different tasks that aren't working memory. So it's not just a working memory network, but it's some type of function that actually takes a lot of comparing different tasks, different conditions to come up with a description of what that network's doing that fits all of these different situations. There are types of fMRI analysis, task-based fMRI analysis methods that are univariate. So in other words, if you have 25,000 brain areas, every one of those areas gets its own test. And so that's like a standard SPM analysis, an AFNI, and the standard type of task-based analyses that you see one sort of blob and there's a difference between conditions. That's a univariate method. So to me, I wanted to be able to look at different areas in that they're combined in different ways. So I'll talk a little bit more about multivariate methods and how we use those. A fairly serious concern for me anyway was the hemodynamic response shape that you put into the model of the fMRI data. So when you have 2 letters, or if you have 4 letters in that task versus 6 letters, then you have to say that, well, what goes into the MRI data analysis is the bold signal and the timing. So you have to give timing about when people did certain conditions and certain tasks. So what is often done, at least in univariate methods, is that a synthetic hemodynamic response shape will be put in at the time that the person did a particular type of the task. So when 4 letters are presented, then there'd be an increase and back to baseline, say about 10 seconds worth of hemodynamic response shape, and then another 4 letters, another increase. So what you're doing is you're assuming that there's a particular hemodynamic response shape involved in that task, but what you find out when you explore the data without assuming the shape is that look how many different shapes there are, and this is not unusual at all. This is a little bit of a long task, so there might be more variety, but you just can't put one shape in and expect to capture all of the activity. That's the second concern. Third thing is that what we do is we constrain the variance in the bold signal to that's predictable from task timing before we do any analyses. That's about 10 to 15% of the variance. So if you're interested in the networks that are involved in doing a task, you can first separate the bold signal into the variance that's predictable from task timing and that which is not. And normally for connectivity type of analyses, that's not done. So I've listed a few here that seed-based functional connectivity, so you take the bold signal and take a seed from that and correlate it with other areas. But you don't, the fact that 85% of the variance that you're looking at, the correlations, actually is not predictable from task timing is not is not a concern. So you can have functional connectivity for resting state, or you can have it for task state, but the difference between those is interpreted without actually extracting the variance from the bold signal that's predictable from task timing. And then there is also methods that involve selecting a few regions of interest. So not a few, maybe a lot, but you're actually starting with somewhere between 25,000 and even 250,000 brain areas or voxels in a bold signal. And it's possible to just explore and include all of those areas. It's not necessary to choose regions of interest, except if there's computational difficulties, so that you just have to, because there's too many voxels to actually do the whole analysis. So there's ways around all of those different concerns. And this is a beautiful movie of the population. This is, Megan found this last week in our lab meeting, Megan Rose, my PhD student. And it is a perfect analogy for what we have to deal with brain imaging data. So using cell phone data, this is Manhattan, and this is Monday, Tuesday, Wednesday, Thursday, Friday, Saturday, and it's actually tracking how many people are at different points in Manhattan. So it's just like brain imaging data, because you have different brain areas in different regions, and you have increases and decreases in activity, but it's actually the location that people are moving to in Manhattan. So here's Monday, Tuesday, Wednesday, Thursday, it's just like different working memory trials. Friday, Saturday, Sunday. So if we use this as our brain imaging data, just looking at, it's like, first of all, if you're doing resting state analyses and the brain's doing these things, you'd have no idea when it's Saturday and Sunday. You don't know, because people are thinking whatever they want to think. You'd be thinking, wow, there's not much activity on those last two. Why not? You don't know. You have to completely speculate. So maybe you'd look at the brain areas, which you can do in resting state, and you say, oh, that's sort of, there's a little bit of activity in Upper Manhattan, so that's where the park is, so maybe that's Saturday, that's the day when people don't go to work. But you're just speculating, right, until you actually prove it. So what you would do if you had the information about the days is you can separate out the days, and it's like having two conditions, Saturday, Sunday, weekend, and work days. And so you can start to think, well, this matters. We know what happens on these days, and we can see what networks are involved. And if you wanted to, for example, know the difference between what part of those population changes are due to tourism and what part are due to going to work, then that's like saying, well, what would you do, right? Because different areas of town are increasing and decreasing together. So you could do a principal component analysis on those data, and then you can correlate the component scores for each time of the day with whether or not people were tourists or whether or not people were going to work, right? So you could maybe take each person and correlate the components that you get over time. But the thing is, you don't have to do it that post hoc way. You don't have to compute the components and then correlate it back with people. You can actually just turn off all the cell phones, people that are going to work, and leave on the ones that are tourists. And that's like taking out the 15% of variance in the bold signal that's predictable from task timing and not looking at the other 85% that's not. So we take just the tourists, look at their phones only, and do principal component analysis on that. That's the whole idea of constrained principal component analysis. And notice that also, if you put in a hemodynamic response shape, you would miss a lot of interesting information. You might just say, we're only interested in increase to noon and then back to 5 o'clock. That's like putting a synthetic hemodynamic response shape. You'd miss a lot of patterns that are, say, something happens at 3 o'clock or the morning. So all of the principles that I talked about, or a region of interest, if you had to take region of interest, you'd obviously miss a lot of patterns too. Just take a few different neighborhoods, and if that's not necessary, you're going to be missing out. So anyway, there's the software, and these are the papers that I'm applied. I mean, you can obviously Google it. It's on NITRC website. There's a publication. Those are all from our lab. There's a few from other labs, not very many. There's a few that use the methodology with animal data, like I showed you before. And Mark already talked quite a bit about this. I'll just quickly go over principal component analysis again. So this is a paper that we published on schizophrenia. So we're a schizophrenia laboratory, and we measure the symptoms of people with schizophrenia, and we have about 60 people in the sample. The thing is that delusions and hallucinations are actually co-occurring to some extent. So some people, if people have delusions, they're more likely to have hallucinations. So there's a correlation between delusions and hallucinations, which is why those two have loadings here in this one component that we call positive symptoms. So positive symptoms basically are delusions and hallucinations. If you measure symptoms of 60 people, those variables will correlate with each other, but not with the other variables. So you end up with clusters of different types of symptoms of schizophrenia, disorganization, and negative symptoms. So that comes from a data set that has 7 variables and 68 rows. So the rows are the people, and 7 are the symptoms that are measured. So that's the loadings that come out of there. I think that's probably a pretty familiar type of output for you. When we have fMRI signal, you might have 25,000 voxels, and over time, so you'd have 300, in this particular example, we have 300 scans per subject, so the whole brain scan is taken once every 2 or 3 seconds. And we have 10 subjects, so we stack those all one on top of the other. So 300, 300, 300, and then the number of columns, the number of brain areas, and we can morph all of the data and warp it into the same space so that people have roughly the same, one column of data can apply to all people. And that's the way we set up what we call the Z matrix. So now Ryan will be able to show you how to load your own Z matrix. Okay. So can you, and I'm going to ask him to do certain things. So can you just, can you,
so everyone knows, you can get the data and all the code from my GitHub up there. I think a lot of you already have the data, but the code is up there in Jupyter notebooks, and it's in Python, it's in MATLAB, just an M file, and it's in a MATLAB live script. So if you want to run it along, or if you want to look at it after, it's going to be up there.
Can you show them where the files, show what the scans look like in the directory?
In the directory?
Yeah.
Okay, so basically we're just going to go through here, and the scans are just going to be basically files in the directory. So I can do, let's take a look at that, and then I'm going to do ls image path. And so these are the scans.
Yeah, can you, how many are in there? 300? So
214 scans.
So each one of those has been normalized to a template, and that template can be used for all subjects. So this is only one subject, and there's 240, right?
There's only one subject, and there's 214 scans.
214, thanks. So each one of them, that actually, these data were collected on a 1.5T scanner, and the scans are 3 seconds. So there's 3 seconds worth of data. That's how long it takes to scan the whole brain. And each one of those files is 3 seconds worth of time. So we're going to take each one, read it in, and just start stacking them one on top of the other so that they run from top to bottom. So I think Ryan has a code to show you how to do that.
Yeah, so I'm just reading in one here, and I'm just showing you, yes, this is an actual brain image. It looks like a brain. And then I'm just here, I'm just reading them all in. I'm using a MATLAB cell fund function. So basically what that does is it applies the same thing to all the things in Accelerate. And so you can do that, and it's going to load all of them in. And you can see that if I print out scan header, I'm going to have 214 image headers. And then I'm going to read in all the images, and I'm going to have a matrix full of 214 rows, and each row is going to be one of those images, and each image has 65,000 voxels in it.
Is that things that are completely already known, or does that help at all? So, sorry, any questions? No, okay. Okay. We'll get to the other part in a second. And I think I'm here, right? Yeah. So what he's done is he's read one subject worth of data in. Here I have 25,000 because you can select 4x4x4 voxel or 3x3x3. He had 3x3x3, so it's 65,000. I have 4x4x4 in these data, so that's 25,000. So principal component analysis, the loadings are what you put onto the brain image. So the blue, basically that brain image is a template. It's not one person's brain image. And you just cut off the magnitude that's displayed. So those are component loadings. And the negative loadings are in blue, and the positive loadings are in red. And we just don't show anything when they're not above a certain threshold. And that would be, now, if you did the principal component analysis like this on those data on a lot of subjects, presumably you get out some networks that are not optimized to tasks, because even though a person was doing the task, as you know, 85% of the variance is actually not related to the task, or at least is not related to the timing of the task. It might be related to doing the task, trying to stay on focus, that just things that aren't related to when you actually presented the stimuli. Okay, so constrained principal component analysis is a general framework that combines regression and principal component analysis into a unified framework. So Yoshio Takani developed this. He's retired now. He lives in Victoria now, and that's how I met him. My supervisor from University of Victoria, Yoshio, lived there in the summer, and we'd sort of eat sushi and drink beer and talk a lot about multivariate statistics. And he is a world-class psychometrician. And CPCA is much, much more than what I'll talk about today. I have some slides that explain the whole thing, but won't make you suffer through those. But anyway, we've known for a long time the potential of CPCA, but really it's taken 15 years just to publish papers on the simplest method. We're starting to use a more complex model now, but it's never been published. Anyway, constraints on rows and columns of data. So Yoshio's developed methods to simultaneously constrain rows and columns of data, and you can extract components, or it uses regression to orthogonalize the data into multiple sources. That's the basic idea. So in this case, we're going to constrain the bold signal into what is predictable from task timing and what is not. So where do we get the task timing? You have timing vectors that go into all functional MRI that is task-based data. So what goes in is actually the bold signal that Ryan just read in, a mask, which we use that we do all the pre-processing with SPM. SPM will give us the normalized data. It also gives us a mask. I think actually we do our own. I think we use its code to compute a mask, but the mask just tells you which are gray matter, which is brain, and which is just air and bone and things like that. So if we have a mask and we have the bold signal and we have timing, that's actually all we need. So now we're going to talk a little about the model, which is very important. And first of all, I think Ryan has some code that will show you what the synthetic hemodynamic response shape looks like. So if you were to make the mistake, the grave mistake of assuming a hemodynamic response shape, this is what often people use in their model. So you can actually separate the bold signal into what's predictable from a hemodynamic response shape and what's not. We don't do that because we don't assume that shape. We use a finite impulse response model, but Ryan will just show you what that would look like if you were to use it.
Yeah, so basically, this is really similar to, I don't know if anybody's used SPM. SPM does this a lot when you're creating the GLM models. Basically, what it does is it takes the event onsets and it makes convolution with a sort of predicted HRF shape, and just right there. And basically what that's going to do is you're going to take those all together and you're going to append those and make those into a predictor matrix.
Can you show them some examples of timing vectors?
Like just individual one of these?
See, yeah.
Okay, yeah. So one of these is going to be a lot of zeros where you're not predicting any sort of response, but when you actually do have an event here, for this condition, basically the first event was pretty far down, so you can see it's about 55 scans in, and when you do, you kind of have a predicted response shape when you actually get to that spot.
Can you show the actual timing vector? The one that just has the number 55 for the...
Oh yeah, the timing vector is just up here, so right. This is the one that we're looking at.
Yeah, so that's got 2, 4, 6, 8 letters. So in this study, we didn't just have 4 and 6 letters, but there were 2, 4, 6, or 8 letters. So that is the other piece of information. Besides the bold signal and the mass, this is all you need. So the reason he showed all zeros and then some numbers at the 55th scan is because that was the 2-letter condition. So the first time that condition came up was at this 55th scan, which times 3 is about 150 seconds. Anyway. So do you have the fur model stuff there too?
Yeah,
we'll just flip back to mine for a second, is okay?
Oh yeah, that's fine.
Okay. So we don't use that type of model because, like I already said, we don't want to assume a shape. We use a finite impulse response model. When the finite impulse response model works a little differently. So in this case, 4 letters were presented here. So we start estimating the increase in the bold signal right on stimulus presentation by putting a 1 in this column. So this is subject 1, load 4, so 4 letters, scan 1. So the very first scan when the 4-letter condition is presented. So put a 1 there. So we want to estimate the change in the hemodynamic response shape just on the very first scan when it's presented, which is not going to be anything because it takes time for the hemodynamic response shape to start to increase. That's why we also estimate another column here for the second one, third one, fourth one, fifth one, sixth one after the stimulus presentation starts. So the hemodynamic response shapes that I showed you at the beginning, there's going to be 1 column for every time point after stimulus presentation. So if we have 10 time points, we have to have 10 columns in this model. So that's what we call the model. The FIR model is in this model. So Ryan will just show you some code for how you create a FIR model. So you get the timing, and then there's a little bit of code that creates the type of model you'll need for that set of timing vectors.
Yeah, so for creating the FIR model, it's really not that exciting, basically. As you just saw, it's 1s and 0s. You do have to think about and make a prediction for how long you think the signal is going to last. But other than that, what I'm doing right here is I'm taking each condition, and if there's an onset, I've decided that we're probably going to see a signal for about 8 time bins after that, or 8 scans after that. And so basically, when I see an onset, I'm going to take that scan, and then the next 8 scans after that, and I'm going to put a 1 in the model. If not, I'm going to put a 0 there. That's basically it. Yeah.
That's it?
Yep, that's basically, that's all.
Okay. Hopefully you all had a chance to try that if you wanted to. Okay, so now we have, look at that. Remember that from when you learned about multiple regression? Data equals model plus error. So the dependent variable equals the independent variable plus some error. So in other words, the dependent variable is a combination of the predicted score and the error score. Does that all sound familiar? So the only difference, of course, that this is 25,000 variables. So there's 25,000 predicted scores here and 25,000 error. It's just that the variance, 100% of the variance has been split into about 10% here and 90% there. But they're still the exact same matrix sizes. So that's just a multivariate multiple regression. So in this case, again, about 24,000 voxels. You get the 24,000 voxels in this, G times C. So C is actually the regression weights. So there's 960 columns in G, which is the FIR model, for some combination of the conditions, the subjects, and the time bins. So every combination of conditions, subjects, and time bin gets their own column. And so G times C, so this is just a set of regression weights for those 960 independent variables for every one of 23,621 dependent variables. So this has nothing to do with PCA. This is to do with multivariate multiple regression. And this is beta weights. This is the independent variables. So the independent variables times the beta weight is the predicted scores for 25,000 dependent variables. Pretty straightforward, right? Nobody's nodding, and so I don't really know if it's straightforward or not. Okay, so if you can do that, which is the constrained part, now you can do PCA, or you can do ICA, or you can do whatever you want to do. But the point is, you've now gotten the important 15% out. So singular value decomposition is principal component analysis, more or less, with some scaling, some tricks in scaling. So with singular value decomposition, you get a left singular vector, a right singular vector, and some singular values. So singular values are rescaled eigenvalues, or maybe eigenvalues are rescaled singular values. These are basically component scores. These are basically component loadings. And you can put those component scores onto a brain image. Component loadings, you can put them on the brain image like I mentioned before, but now those are going to be networks that are specific to the variance predictable from the task. Not just resting state or some type of task state with 100% of the variance left in. Then these are actually very important because they give the value and the activity of each one of those networks on every single brain scan. So if you find out the numbers that would predict those component scores from the model, you get predictor weights. And there's a predictor weight for every time I give this part of the talk, I can tell nobody can tell. If anyone can tell what I'm talking about, put up your hand. I know all my students know exactly what I'm talking about. Nobody else. Thank you, one person. It's not that, for some reason, this everyone can kind of get, but for some reason this people cannot get. And it's really, really not that difficult. I guess the problem is you have to understand what's in the model. So if you took the component scores, there's 3 of them, and you take one component score, and that's your dependent variable, and then you use your independent variables and you get beta weights. So what are the weights that, when applied to that model, produce the component scores? Those are the predictor weights. And those, believe it or not, create hemodynamic response shapes because there's a higher weight for the columns of G that are close to the peak of the hemodynamic response. Believe it or not. It works. Are we going to show them how to do that?
Yeah.
Okay, so anyway, you can do your own fMRI CPCA right now with like about 4 lines of MATLAB code.
Yeah, it's really easy with MATLAB. MATLAB has a lot of stuff that's kind of built in to do all the stuff for you, and I'm just going to make sure that I've standardized my G matrix. And so, yeah, this line, this basically slash does regression for you in MATLAB. It's the ML divide function. It does a lot of kind of fancy stuff about what functions to do in the back end, but basically it's a solver for a linear equation, and it will give you the least linear square solution to a regression equation. And that's basically all you really need to do there. You can also use the normal equations, which seem to work faster for some reason for our data, but and they'll basically give you the exact same answer. And then just doing the singular value decomposition like this, you can get the eigenvalues and then eigenvectors, and you can just regress to that and then rescale it, and you'll get the predictor weights. And plotting those, you can see they don't look great because they're for one participant, but if you add more, they will start to look more and more like response shapes.
There we go. So all of the GUI is basically ways to get the data in and out of those. Well, actually, we have to do a slightly different equation because if you have a lot of data, these ones won't work because there's too much memory. So. Is there any more?
No, that's basically it.
Okay, thank you. Okay, anyone want to ask any questions or discuss anything about this? Yeah.
So, understand correctly, the objective of the analysis is to find collective voxels related to some external variable, task, behaviors. To me, it sounds a lot like Hamilton-reduced brain regression model. Partially squared is not.
Yeah.
Things like this. So
Yeah.
what things would you compare and contrast?
Oh, man. Well, first of all, it basically is a cross product of the model and the dependent variable, of the independent dependent variable, which has been used quite a few times in different contexts. But it's not so much, that and doing PCA, basically PCA of the cross product, right? Like what's predictable from sort of taking what's the overlap between the independent dependent variables and doing PCA of that. But it's more the matrices that go into it's not genius math, but it's the matrices that you set up to go into the model that give the information that's interesting. And it's mostly the FIR model, right? So if you think of it as a cross product of the FIR model and the bold signal, and then principal component analysis of that type of, that's basically it. So it's not that difficult. But it's the interpretation that's difficult, because it's the hemodynamic response shapes that you get out from the FIR model that are what we spend all of our time looking at and trying to compare across experiments and come up with a function of the networks. But we're also recognizing that there's a limited number of networks, or at least there's only about 10 or 11 classes of networks that come out consistently, which you wouldn't, unless you explored like this and allow the hemodynamic response shape you don't put in any assumptions about the hemodynamic response shapes and allow it to tell you what the hemodynamic response shapes and what the networks are. We're starting to realize that they're not the resting state networks, but they're task-based networks, and they're consistent. So, yeah, basically that's it for the math. I mean, just what you've seen there. But it's the output, that the way we set up the matrices that gives us the information that helps us come up with these interpretations, functional interpretations of the networks. And so, this is what I was just actually saying. So resting state networks are well determined, and like I've already mentioned, some people have already talked about, that's actually very replicable, and we have names for them. Like I said, some of the names are a bit speculative and maybe making assumptions of what those networks are involved in, like dorsal attention network. So the task-based networks are not the same, and this is a physicist that here from UBC that won an award for basically having a temperature chart of the universe, and he is totally happy with saying this is just an image. This is just turning the raw data into an image you can visualize, which is what resting state networks are. It's just a visualization, right? But let's start to interpret, and that's why the hemodynamic response shapes are so important, because it's the interpretation that's the difficult part. It's not the imaging that's the difficult part. So now we're trying to figure out what the function is of these networks. So how much more time do I have, or how much more time can we handle? It's 4 o'clock now. I'll make it shorter than that. I'll make it shorter than that, because this is really the part that is frustrating for people that don't care about tasks, because this is going to involve trying to basically link up the hemodynamic response shapes to the different tasks. And we found that combining tasks is very, very powerful. So I don't want to make you, I know it's late in the day, I don't want to make you suffer through all this, but I just can't help myself, so here we go. And by the way, Ryan, if you want to go sit down, that's, I think, all we have for that thing. There's more people looking at you. Thank you. Okay, here's Nicole Sanford. She's right there. And she's been working on combining. So let's talk a bit about the response network. And come on, just pretend it's a resting state networks. Then maybe you guys will be more into it. This is new a little bit, task-based networks. The response network is actually one of the rest. I think you can find that in resting state. So this is not new. But I already showed you the shape that should be for this task, 4 or 6 letters, delay, then a response. Sometimes there's a 4-second delay, sometimes there's not, so you have a 4-second delay between the 2 peaks. We saw that, plus no difference between 4 and 6 letters. So then Nicole combined this with this other task that we have in the lab. So in this one, you see a table, and you think to yourself, something you eat breakfast on, or you hear a sound file play that says something you eat breakfast on. Well, I mean, a person might not think exactly that, but that's the idea. So the thing about that task is there's no response, right? So if this is really response network, and I see a hemodynamic response for the other task, I'm wrong. So you can test hypotheses about the functions. This is a really easy one. And so here is those 2 tasks combined. So you remember the matrix of bold signal. You have a matrix of bold signal for task A and all the people that did task A, and then a matrix of bold signal for task B and all the people that did task B. So different people that did the working memory task and that did the thinking to yourself task without a response. That's not optimal. Ideally, you'd like to have all the same people in both tasks, and maybe it'd be better if you design the task so that you're manipulating into something instead of, but there's so much fMRI data out there that you can quickly test hypotheses like this in a couple of days. And so we're starting to do that just to sort of, confirm the interpretations we're making. So there's the 2 peaks, no difference at all between 4 and 6 letters, and look at the one where there's no response. Not only is there not a hemodynamic response shape, there's some type of suppression going on. So that's a first hint that this is that this sort of supports our hypothesis, even though we might have thought it would be flat. But if there is some type of suppression of the response network when you don't respond, that's not exactly completely off interpretation. The big problem would be is if it also was active, and so that gives us something to work on. And then this is another task. This is a Stroop task, and in this task you have to sometimes name the color. So in this case, you say yellow, blue, red, and here you read the word green, red, blue, and here you actually name the color. So this is going to be green, red, blue again. So this involves some type of cognitive control. It involves some word reading, but it definitely involves a response, and it involves a 2-handed response this time. The last one was a 1-handed response. And this is the task with no response. So in this case, we get a bilateral type of activation, and we don't see much differences between conditions, although the hardest condition is a little bit higher. and then we don't see any response at all in hearing and generating. So these are the type of hypothesis testing we can do by combining experiments. This is a language network, so this is a lateralized network here. This is Wernicke's area and Broca's area. We get a lot of visual cortex on here, but that's probably just a combination with visual network because they are reading in this. Oh, wait a minute. They're at least looking at something. And so then we combine that task. This is a thought generation task that we just talked about, where you're thinking to yourself or you are hearing a sound file. And this is an oddball task, and this involves just hearing beeps, and when there's a high beep, you press a button. So there's no language involved in that task, but there is in when you're thinking or hearing the sound file. And we find a response in the hearing and generating conditions, but there's no response, no real response in the oddball task when there's no language. So again, testing hypotheses about the function of these networks. This evaluation network is actually consistently something that happens after the response, which you wouldn't find if you put in a hemodynamic response shape as an assumption. We call it evaluation because under a number of conditions we find it's involved when you're thinking about what you just responded, when it's a difficult situation and you're thinking, was that right or was that wrong? So this is word reading when it's just black ink. This is color naming when it's just X's. So in other words, there's no interfering word. And this is incongruent color naming. So you have to say red ink when it's actually blue. The word says blue. And you can actually get slowing when you're doing word reading when there's an interfering color as well if you switch tasks. And this is hearing and generating. And we see activity when there's incongruent, but not when there's neutral. So when the task is more difficult, after the response, there's activity in this network, but not during the task when there's time to respond. So it's this process of thinking, what did I just do? So when it's incongruent word, you say, did I say word? Was I supposed to say word or color? That type of thinking process. So again, there's a number of different tasks we're using to look into that. So in this task, you'll see there's sort of something that looks like an umbrella. And so you'll say, yeah, that's an umbrella. But then you get more information. You fill the picture in, and it actually turns out to be a bat. So this is disconfirming evidence. So this is when we tie into schizophrenia, delusions are a situation where you believe something strongly, but you want nothing to do with undoing that belief. If you ever talk to someone who has a delusion, typically you can't talk them out of it because they're just set. So we study how to integrate disconfirming evidence a lot. And this is a network that's involved in disconfirming evidence. So when there's more activity when you disconfirm than when you confirm, and it turns out to be the same type of network that I showed you earlier. Okay, I won't make you suffer through all of this, but I mean, we just have, sometimes combinations of the networks come up. Like in this case, this is the auditory oddball, and so you're hearing the odd tone and you press the button. So that's why you have superior temporal type of activity here and some response areas on the network at the same time. And I have the hemodynamic response shape for this one. This is the oddball, so you're pressing when you hear the odd tone, but this is the one when you're not pressing. And it's a little bit higher in the condition when you're hearing than when you're not hearing. So it fits with these hypotheses. So then you have to think about why is there more activity in the oddball, even though you're actually there should be more activity in the hearing condition. So it may have something to do with the difference between linguistic and non-linguistic. But we have to try with a lot more tasks. Okay, let's go enjoy the sun. That's about it. So I already introduced you to Ryan. My graduate students, Nicole, Jessica, and Megan are up there. And Christine Tipper and Mahesh Menon, my close colleagues here, and of course Yoshio Takani, who developed all the CPCA. Thank you.